Okay, I need you to tell me if you cannot see the picture of this animation. Okay, أنا حجرب حطها وأنتوا إذا ما شفتوا بتقطشوني دغري أو إذا بس مبين الصوت without the picture.
For the process to work correctly. So what's something great about being mRNA?
You can see the screen?
Yes.
Okay.
Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You could see on a codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, It couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein
Okay.
structure and function.
Yeah, this is it. هدي كان summary عن كل شيء نحنا أخذناه امبارح عن initiation, elongation and termination. Okay, before we watch the next animation بس خلينا نشوف who joined. أمل, سارة, we have Mia, and Wissam. Yeah, let's watch the second animation.
But the process
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will
Okay, the poly A tail هي المطرحة that is not translated. Okay, هي مش a part of the reading frame. هي يعني ما قبل عندي أو ما بعد ال AUG. اوكي او الستوب كودون. فاذا هن المطارح اللي هي ما برا what is beyond the reading frame يلي هي from AUG to a stop codon.
code for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches.
فاذا كمان انا عندي هون شيء for protection. هودي مش مطلوبين تفاصيلهم. اوكي، وهلا عم ببلش عندي وبنهايه المسنجر RNA كمان انا عندي هيدي البولي A tail كمان it is for protection from the enzymes that are found in the cytoplasm. أنا اللي بيصير عندي هون ببلش الريبوسوم scans to find the first AUG. As you can see,
attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, A release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Okay. So this is the second animation. It shows the steps of translation. Finally, ننهي.
DNA is very long and cumbersome. The strands of DNA also code for many different proteins. To get the code for the desired protein from the nucleus to the ribosomes in the
Then this shows the transcription process. هيدي translation process. Okay, فإذا هيدي حتلخص لي transcription translation.
cytoplasm, a copy of the small necessary section of DNA will be made. The copying process is called transcription, and it occurs in the nucleus. It's the first step in protein synthesis, the process of making protein. The second step is called translation. A portion of the DNA is unzipped so that the mRNA can be made from the DNA like a template. That's transcription. Nucleotides of RNA match with one strand of DNA and make mRNA. RNA polymerase unzips the DNA and puts RNA nucleotides into the right place. C and G nucleotides match up, A and T nucleotides match up, but anytime the RNA places a complement to an A on the DNA, it places a U instead of a T in the RNA. Remember, RNA uses uracil instead of thymine. So, if the DNA strand looked like this, the complementary mRNA strand would be the complement, but each time there would normally be a thymine, there's a uracil in its place. The mRNA also doesn't copy the entire strand of DNA. It only copies the portion of DNA that codes for the protein it wants at the time. In fact, most of DNA doesn't code for anything. Only about 2% of it codes for protein. So the small section of coding DNA has a start and stop signal. After the mRNA is formed, it moves out of the nucleus through nuclear pore and goes into the cytoplasm. Remember, there are 3 different types of RNA. The purpose of the mRNA is to get the protein code out of the nucleus without pulling the DNA out of the nucleus. Then the mRNA will need to find a ribosome in the cytoplasm. Ribosomes can be found on the rough ER or floating in the cytosol. mRNA is read 3 bases at a time, and these 3 bases are called codons. Now, the ribosomes are made of protein and RNA, and the ribosomes are the protein-making machines that read the mRNA code and add the correct amino acid using tRNA. Remember, tRNA stands for transfer RNA, and that's because its purpose is to transfer the right amino acid to the ribosome to build the protein. The tRNA has an anticodon on one end, which will match a specific codon on the mRNA, and has a specific amino acid on the other end. Together, these 3 parts will make translation happen. Translation occurs in the cytoplasm of the cell wherever there's a ribosome. To get translation started, mRNA attaches to a ribosome and a start codon must be read. It's usually AUG, and I remember that school starts in August, and that helps me remember AUG. The first amino acid is brought in by tRNA. The anticodon on tRNA matches up to the codon on mRNA. Then the next tRNA molecule moves in and matches up with the mRNA codon. This time, the amino acids form a peptide bond and link together. Then, the first tRNA can detach, and the mRNA shifts through like ticker tape, and the next tRNA molecule can come in. The protein grows until a stop codon is reached.
Okay. Yeah.
Once the stop codon is
اوكي حننتقل هلا لآخر فكرة بالدرس. اوكي موجودة بنفس الصفحة page 57. It's called Apollosome. This figure is called Apollosome. Apollosome is the presence of many ribosomes. وجود كتير ribosomes on the same messenger RNA. This is a ribosome, this is a ribosome, okay? The presence of many ribosomes on the same messenger RNA, it's called a polysome. As you can see, if I have, let's say, if I have 10 ribosomes. إذا أنا معي 10 ribosome are moving along the same messenger RNA. Okay? Do you think, or how much is the number of the protein synthesized? How many proteins will be synthesized? إذا أنا عندي 10 ribosomes عم بيمشوا على نفس المسنجر RNA، كم بروتين حيتصنع عندي in this case? Do you know?
No.
Okay, طيب let's say each ribosome can synthesize one protein.
10, yeah.
10. So if I have 10 ribosomes moving along the same messenger RNA, إذا أنا حأحصل على 10 proteins. Do you think these proteins are identical or different? أنا عندي نفس المسنجر RNA، نفس الكودونز. مفترض كل رايبوسوم يعطيني a different protein ولا الرايبوسومز كلهم بده يعطوني نفس البروتين in this case؟ نفس البروتين. إذا whenever I have 20 ribosomes, the number of ribosomes is equal to the number of the protein synthesized. I will obtain 20 proteins. إذا عندي 100 ribosome بيطلع عندي 100 protein. وإذا الريبوسومز in the case of polysome نحن نقلها polysome أو منقلها حتى polyribosomes. Poly means many. So polysome or polyribosome, many ribosome on the same messenger RNA. إذا أنا عندي 100 ribosomes moving along the same messenger RNA, I will obtain 100 proteins, but these proteins are identical. They have the same amino acid sequence. نحن بنعلم هيدي الرسمة اللي موجودة page 57 document E. It's the polysome. Okay, it shows the ribosomes. شوفوا كيف السيكونس، كل ما بيكون الرايبوسوم ماشي أكتر على المسنجر RNA كل ما بيكون السيكونس of the peptide is bigger. Is it clear? Is it clear, miss?
Yes.
Okay. Finally, لازم تعرفوا إنه أكتر step بالبروتين synthesis ما بين الانيشيشن الالونجيشن والترمينيشن هي الالونجيشن that is needing the most.
طالع صوتي.
Yes, we can hear you. Yes. Okay. هي elongation it needs the most amount of energy. أكتر amount of energy needed it is by elongation, because in elongation we have the peptide bond and the translocation of the ribosome, formation of peptide bond. The translocation of ribosome, they need a lot of energy for them to occur. إذن أكتر خطوة بدها طاقة هي خطوة elongation, because it has the translocation and the peptide bond. Okay, أنا ما بدي أتأخر كتير لأنه عندكم كتيبة. If you have أنا باقية معكم. If you have questions you can ask. يلا start writing, miss. I'm with you.
هسه أي صفحة على الكتاب؟
57. Page 57.
اوكي